Hello, everyone. I'm Roland Dunbrack from Fox Chase Cancer Center. And this is, well, welcome to my kitchen. This is the lecture on structural bioinformatics in BMSC8203. The way I'm going to do this is share my screen here. This is Zoom, and go through the slides, and you'll see me in the upper right corner. I do use my hands occasionally to describe something. So you can kind of watch both. This is going to be about an hour and, I don't know, 15 minutes maybe, lecture and with the slides, and I'll tell you where those are. And then there'll be a separate video on how to use PyMol and the PDB's website and some other websites for doing protein structure analysis or protein structure prediction or sequence analysis. This is the second time I've recorded this video because the first time I did it, I forgot to hit the share screen button, which is right there. And so it was just me talking for an hour and something with no slides. So let's do it again. Hope everybody's doing okay and that you're at home and trying to keep healthy and keep everybody else healthy. So I'll put these slides at these two website addresses, both the PDF and the PowerPoint. I asked Dr. Engel, I think, sent you a link on how to get PyMol. And so this was the link, and hopefully everybody's done that and installed it. But you click this link and it asks you for a password, and it's June 2020. It's the name and alpha helix. And you get to this page, and under here where it says PyMol 2.0, you're actually going to get PyMol 2.3. There is this file, which is the license file, and you just want to save that either to your desktop or wherever, or your applications folder, wherever you want. And then hit the second button, which says installers. And then just follow the directions, these installation instructions, either the Windows exe installer. If that doesn't work, try the zip archive, and PyMol should be inside that, or the disk image or Linux tar file. And then once you open up PyMol, it will ask you to browse to the license file, so wherever you put that, and that will allow you to use it, I think, through the end of June. But you can always get another student license that's probably indefinite while you're still a student. Okay, so let me close all of that. And if you have any problem with that version of PyMol or somehow it didn't install, we had trouble here at home trying to get it onto Windows properly. These are the old versions, version 1.8. There are some features that it doesn't have, but they're sitting on our website, and these don't require an ongoing license. So if you can't get 2.3 to work, then try one of these. Okay. What I'm going to try to do is sort of go through some different aspects of protein structure in a bioinformatics kind of way. I know you probably all know something about protein structure, but this will get into some details that maybe you haven't seen before. This is the outline. We're going to talk about, very briefly, different ways of determining structures, experimental structures, and then go in some depth in dihedral degrees of freedom of the main chain and side chains in proteins. And then talk about hydrogen bonds and protein folding. And then beta turns in loops, which is something that we've done some work on recently. And biological assemblies. If a protein is in a oligomer in a crystal, which oligomer is it? What does it look like? Issues of symmetry. Okay, the structures in the PDB, there are three main ways in which the structures have been determined. The first of those is X-ray crystallography, which means purifying a protein, concentrating it in a particular buffer so that it will crystallize, and then taking that to the synchrotron and shooting X-rays at it, and you measure a diffraction pattern, kind of spots. And from that, you also need a guess of what's called the phases. They're angles associated with each spot in the diffraction pattern, but you have no idea what they are to start with. You can get a good guess if you know a homologous structure of a homologous protein. Another way to do it is to dope the crystal with heavy atoms. There's a long history of different ways of getting an initial guess to the phases. With the spots and the phases is a mathematical transformation, Fourier transform, that you can get an initial guess of what the electron density looks like. Once you start looking at that, you can begin to see alpha helices and side chains, and you start putting in the coordinates. From the coordinates, you can calculate the phases, and then go back and add the phases to the diffraction spots, their intensities, and then recalculate the electron density. It's just sort of a cycle. Keep adding to the atomic model, refining it, getting better phases, adding the experimental structure factors, those are called the spots, their intensities, and getting better electron density. The second way of determining structures is nuclear magnetic resonance, or NMR. What you're getting from that is the distance between atoms in a structure. And so you can reconstruct the structure by, I'm just seeing with them. Yeah, okay. You can reconstruct the structure by knowing the distances between atoms. And this are usually done by kind of simulation, simulated annealing, where you use those distances as constraints, usually between residues that are in contact with each other to fold up the protein. The third way of doing it, which has increased rapidly in the last few years, is cryo-electron microscopy, or cryo-EM. This is especially good for large complexes. Doesn't actually work that well for small proteins, which is the converse of NMR. NMR works well for small proteins, does not work well for large ones or assemblies. In cryo-EM, the protein is purified and then vitrified, make a sort of very thin layer at sort of cryo temperatures, and very thinly sliced. And then the electron beam is passed through that sample, and what you see is basically a projection image. And you can collect millions of these, and then cluster them by what sort of orientation they are, because they'll be all kind of different orientations on that surface. And from each cluster, begin to kind of superimpose those, which will increase the sequel to noise ratio, so that you begin to see what look like alpha helices and beta sheets and side chains, and then you can build a model. And again, it's sort of a cyclic type thing that you get, you know, a better model that fits the electron density. Cryo-EM has undergone kind of a resolution revolution. It used to be that you couldn't do a structure better than, you know, 8 or 10 angstroms. Now you can do them down to maybe 2 angstroms. Most of them are still in the 3 to 4 to 5 range, so they're kind of like low resolution crystal structures. The other thing is that the resolution is not uniform. Sometimes parts of the structure don't, or parts of the big assembly just are moving around too much, and it can be very hard to get good electron density for maybe one subunit, and other subunits look fine, which is not really usually the case in crystallography. And then sometimes people combine both of them. If there are structures of the individual proteins in some big assembly, those can be used to model the electron density in a large EM structure. Okay, that was just to take a look at the experimental methods. I'll show you some PDB pages later about how those are annotated in the PDB. But a main topic in this lecture is going to be about dihedral angles. And I think most of you probably know some organic chemistry, and you know that, you know, bond lengths of atoms, particular kinds of atoms, particular kinds of bonds, are usually pretty fixed. There's a little bit of vibration around them, but, you know, every example of a carbon-carbon bond or carbonyl, carbon-oxygen bond in a protein is basically the same length. They can vary just a little bit. The same thing is true of bond angles. If you have, you know, the C alpha atom, the carbonyl carbon, and the carbonyl oxygen, that bond angle is probably around 120 degrees and doesn't change much. And so that's not really a degree of freedom in a protein, or it's a very minor one. But what does change a lot of the dihedral angles, that's the angle that connects 4 atoms in a row. And it's the plane that's determined by the 1, 2, 3, first 3 atoms. The angle between that plane and the angle that is atoms 2, 3, and 4. So this part of it, those two triangles, I guess. And that's 0 degrees where they're parallel. And then this would be 90 degrees towards me, or minus 90 degrees that way, and that's 180 degrees. The way to remember it, the way I like to do it is looking, as you see in this image, of moving the atom in the front. So if you're sort of looking at it like this, moving the atom that's closest to you, this is closest to me. If you think, from high school algebra, polar coordinates, where you have a radius or a distance from the origin and an angle that starts at the x-axis, and you should go counterclockwise, that's a positive rotation. So 90 degrees, plus 90 is the positive y-axis. And so if you think of that atom in the front going in that positive direction, that's a positive dihedral angle. And if it goes the other way, it's a negative dihedral angle. I just use that to help me remember. For the protein main chain, there are 3 dihedral angles that are important, and that will help us build the entire chain. If we knew what omega, phi, and psi are, these 3 dihedral angles, we could build up the entire protein. Might have to add in a little bit of bond angle variation here and there, but for the most part, that will describe the whole protein structure. Omega is the for a particular residue, we're going to do this red residue in the middle, is the C alpha atom of the previous residue, carbonyl carbon, then the nitrogen and C alpha of the residue that we want omega of. These atoms, Ca, Co, N, and Ca, are basically in a plane. And that's because there's sort of a partial double bond character to this CN bond. So any deviation from that would be, there'd be an energetic penalty to that. So it's sort of very flat like that. For all amino acid types except proline, 99.5% of the residues in proteins, this angle is 180 degrees. So it's like this. In proline, which is a ring structure that is N, C alpha, N, C alpha, C beta, C gamma, C delta, back, and then a covalent bond back to this nitrogen, when there's no hydrogen. That ring structure, since you've got C delta and a C delta in kind of the same distance from that nitrogen as that C alpha, looks a little bit more symmetric. And so what you can get is what's called a cis peptide bond, which this C alpha C and C alpha dihedral, this omega, is 0 degrees and not 180 degrees. That's about 5% of prolines. And like I said, it's maybe 0.5% of other amino acid types. It varies. At lower resolution, sometimes people put in cis peptide bonds in residues that are not proline, and they're incorrect. There's just no justification for it. On the other hand, sometimes it's under-annotated because people don't expect them, and so therefore they don't put them in the structures when they actually are there. It's a very difficult thing to annotate at lower resolution. The next dihedral is phi, which is the carbonyl carbon of the previous residue, and then NC alpha C of the residue in question. And then psi, which is NC alpha C of the residue, plus the nitrogen of the next amino acid. And so those 3, just omega phi psi, omega phi psi, omega phi psi, you could build all of the atoms of the backbone. If we look at the side chain degrees of freedom, those are labeled chi1, chi2, chi3, chi4. Depends on the length of the side chain. Some of them, short ones only have chi1. The long ones have up to chi4. The first one, chi1, is NC alpha, C beta, C gamma in most side chain types. The C beta is basically fixed. If I know NC alpha and C, I can place the C beta really easily at a sort of fixed position. And so the first degree of freedom is, where is that C gamma atom? The C gamma atom could go around in a circle like this, 360 degrees. So chi1 tells you where the C gamma atom is. I know NC alpha, C beta. I just need bond length, bond angle, and that dihedral chi1, and I could place C gamma. Chi2 will help me build the C delta. That's C alpha, C beta, C gamma, C delta. So it's the rotation around this bond, but that helps place this atom. Remember, this is sort of like that. Chi3 gives you the C epsilon, and the C delta, chi4 gives you the N zeta atom. This is a long side chain lysine. These are all labeled after Greek letters. So alpha, beta, gamma, delta, epsilon, zeta, eta is the next one. If we go back to backbone dihedral angles, this is called the Ramachandran map. It's a plot of phi on the x-axis versus psi on the y-axis. And you see that the population is not uniform. There are places where there are just no amino acids have that combination of phi and psi. And that's because there are steric conflicts. Atoms either with the C beta atom with the previous residue, the next residue, there are atoms that just at certain phi psi combinations, the atoms are colliding and you can't have those conformations. There are all sorts of different nomenclatures for this. For a lot of purposes, we use a really simple one that spells out ABLE, able, where this is the A region. And it's named that because alpha helices would be right here at this very lower right corner. But in loops, sort of regions in between beta sheet strands and alpha helices, there are a lot of residues that are not just in that corner where alpha helices are, but actually in a very large region like this. And so we name that A for alpha, the alpha region. Beta sheets are in this upper left corner. And so sometimes we label this whole segment as the B region. But you can tell there are actually subpopulations in that. This big one here is called polyproline 2. And it's named that for polyproline helices that would have a phi psi repeating pattern in these values of phi psi. Those are not that common, but there are a lot of residues, particularly in beta turns, that have conformations in that region. So we just call this piece the P. L is for left-handed helix. It's this region here. There really aren't left-handed helices in proteins, but if you repeated this dihedral pair over and over and over again, you would get an alpha helix that is left-handed instead of right-handed. And so typical alpha helix is right-handed, so this would be left-handed if it existed. And then this region down here is E. This is E for epsilon. And most amino acids do not have very many points down there. I'm going to show you in a second. Glycine, which does. Oh, the other thing to notice is minus 180 is the same as plus 180. And so these points down here actually belong to the populations up here. They're just slightly over the line. And that's why we also call this the B region in addition to that. The same thing with these epsilon and epsilons. The phi psi distributions, Ramachandran distributions, differ by residue type. And at one simple level, we can break them into 4 different groups as opposed to all 20 amino acids. This is done by Jane Richardson at Duke University. Every time you see a protein in a kind of cartoon representation with the helices like this and sort of a ribbon, the beta sheets with arrows on them, that was developed by Jane Richardson, and she's done many other very important things in structural bioinformatics. And this is one of them, which is a website called Molprobity, which evaluates structures in the PDB, or you can upload a structure, and it will tell you basically what's wrong with it, whether bond lengths, bond angles, dihedral angles, the side chain conformations, the backbone conformations, are they statistically improbable? And if they are, then there's probably something wrong with the structure. If you've got a bond length of 2 angstroms and it's supposed to be 1.5, the 2 is just absolutely wrong, cannot be right. And it will tell you that. Same thing for backbone conformation. So what Jane's group did is they used very high resolution structures and very careful weeding out of structures that had problems, like atom clashes, and got a list of data points, and then plotted Ramachandran plots like this. The very general case is here in the upper left. This is all amino acid types except glycine and proline. And it's also does not include residues that precede a proline. Those are shown in the lower right. Because they have a different distribution. They have some density that others don't have. And so this is the very general distribution for all 18 amino acids, excluding residues before prolines. And you can see you can see this one is more refined than this very low resolution one that I just showed you. Glycine has only a hydrogen as a side chain and doesn't really have a side chain. It just has a hydrogen atom. And so it can get to conformations that the other amino acids can't, because the C beta atom of all the other 19 amino acids would bump into the backbone in various places. And so you see a lot of density in this lower right corner. You see more density here and it's sort of slightly different position than in the general case. And you see a lot of stuff up here. Again, you don't see anything down there. And it's also kind of symmetric across, kind of, if you go across, take a line from this side through the origin to this side, there's kind of a certain symmetry to it. And that's because you've got two hydrogens on the alpha carbon and no C beta. And so those are sort of the same to each other. You don't have a carbon in one case and a hydrogen in the other, you just have two hydrogens. So you get a kind of symmetry across the origin here. Proline, remember, proline is a ring, and C alpha, C beta, C gamma, C delta, back to nitrogen again. And because of that, the phi dihedral can't vary. It has to be kind of minus 60 plus or minus 20 or 30 degrees. Because if it went any more than that, it would actually break the ring, which would be a lot of energy. And so prolines have this very narrow range of phi. And you can see the populations. Basically, there's two main populations and maybe a small one in here. And they really don't like to be down here. And then finally, the pre-proline residues, proline, because it has NC alpha, C beta, C gamma, C delta bound to the nitrogen, a residue I will bump into that C delta of I plus 1 if it's a proline, which would reduce the population in many parts of the map, including the right side. Basically wipes it out altogether, except just a little bit here. It really decreases substantially the amount of alpha population. You don't see much here. And you get this sort of C beta B region distribution up here. You get this extra bit of density, which some people call a zeta conformation. And so that's this little tail here that you don't see at all, or much anyway, distinct population up here. You don't see it in the general case, you only see it in the pre-proline case. This little thing here is called a gamma, or actually gamma prime. There were sort of little 3 residue turns that kind of stabilize that conformation. That's why you see this little dip here. Molprobity uses these to judge crystal structures or experimental structures. If they have points in these regions that are basically empty, it flags those and it tells you that there's a problem with them. Even the remaining 18 amino acids that are not pre-proline do have different distributions from each other. This is a paper that we did in PLOS computational biology. And so these are Ramachandran distributions of all 20 amino acids in protein loops. So it excludes alpha helices and beta sheets. If we included alpha helices, we would get a really tall spike right at this sort of lower right end of the alpha region here. And it would be so high that it would dwarf the other regions, and you just wouldn't be able to see anything. And we were interested in predicting loop structures, so we did only the amino acids in loops. And there are variations. There are some that have higher L conformations, like asparagine and aspartic acid over here. You can see this left-handed conformation on the right-hand side of the map is, almost as high as the alpha region. That's not true of most of the other side chain types. Glycine, remember, has stuff all over the place, and it is very commonly used in proteins to have an L conformation or this epsilon down here. But there are some that don't like the left-hand side at all. So if we look at isoleucine and valine, they have very little on this right-hand side. And that's because they have 2 gamma heavy atoms, and there's just more ways for those to collide with the backbone in that left-handed conformation, so that it makes that basically forbidden. This paper had a lot of other stuff in it. It had a sort of, how does the Ramachandran distribution vary depending on what the neighboring amino acid type is. So like that pre-proline, but for all 20 amino acid type neighbors to the right and to the left. Okay, if we look at let's say chi1, most degrees of freedoms in side chains are rotameric. And a rotamer means rotational isomer. And so it's just some different dihedral angle values. That's the difference between one conformation of a molecule and another conformation in a molecule. So those are called rotational isomers or rotamers. No bonds are breaking like in other kinds of isomers. And you may remember from organic chemistry that if you have a molecule like butane, which is CH3, CH2, CH2, CH3, the carbons are all tetrahedral. They all have 4 substituents on them. The atoms do not like to have a 0-degree dihedral angle. They do not like to be like this. So 1, 2, 3, 4. Any 4 atoms in a row do not like to be have a 0-degree dihedral angle. And actually the reason turns out to be that there's repulsion of the electrons in this covalent bond and in this covalent bond with each other across this sort of U shape. And so you don't have these kind of staggered conformations. These are called Newman diagrams. If you want to know more about them, you can look up Newman diagrams on Google. And if this was a 0-degree dihedral angle, see it's represented just slightly off just for visual purposes. But if that were 0 degrees, those CH3s, would be too close together and there'd be repulsion between those bonds. Even when it's a hydrogen, so there's another carbon kind of behind this circle. You have to imagine yourself kind of looking down the middle 2 carbon, the covalent bond of the middle 2 carbons, numbers 2 and 3 out of 4. Even if it's CH3 and H, there's repulsion, or even 2 H's. And so even ethane, which is just CH3, CH3, wants, instead of having these are called eclipsed conformations, they want to have these staggered conformations, where if I start at 0 and I just rotate 60 degrees, now if I stay at 60, then they're as far apart as they can get. Because if I go 120, then they're lined up again, because every 120 degrees there's another atom. And so these are the staggered conformations. This one, for instance, is trans, where the CH3 in the atom in the back and the CH2, CH2, CH3 are in a trans conformation like this. If we look at lysine chi1, so the first dihedral angle of lysines, this is called a kernel density estimate. It's like a histogram, but it's smoother and prettier than histograms. If we look at the distribution of chi1 in a lot of experimental structures of lysine, we get 3 peaks, and those correspond to these staggered conformations. So one of them is trans, where N, C alpha, C beta, N, C alpha, C beta, C gamma of lysine is 180 degrees. But there's also the value when it's plus 60 degrees and when it's minus 60 degrees. Minus 60 is the same as 300, just going the other direction. So those are the 3 rotamers, chi1 rotamers of lysine. The staggered conformations are basically in between, sort of around 0, around 120, and around 240, where these blue arrows are. Those are the eclipsed conformations. Those are the ones that are not allowed, and the staggered conformations are the ones that are allowed. This is just what they look like, and you may get, if you look at enough of these like I have, you get used to sort of spotting them. These are what they look like on a structure. So this is for a serine side chain, and remember, if I started at 0 degrees, N, C alpha, C beta, C gamma, O gamma, that O gamma would be kind of this way, would be parallel with this nitrogen. But this is going to be the plus 60 rotamer, so I'm going to go in a counterclockwise direction, just like in a polar coordinates in the XY plane. So move the atom in front in the counterclockwise direction, and go 60 degrees so that I'm in between the N and the C. And that's a plus 60 or G plus rotamer. If I go another 120 degrees, I get to the trans rotamer. And so N, C alpha in the back, C beta, O gamma is like this, so it's trans. And then if I go another 120 degrees, I get to the 300 degree rotamer, or the minus 60, and we call that G minus, gauche minus. By the way, trans is labeled for the Latin word trans. Caesar, when he was writing about his conquest of Gaul, referred to cis and trans Alpine Gaul. Cis Alpine Gaul, cis meaning the same side, CIS, means that it was the part of Gaul, the Celts that were on the same side of the Alps as the Romans were, on the southern side. Transalpine Gaul were the Gauls that were on the other side of the Alps, and so we use the word cis and trans to mean same side like that or opposite side like that. For the sidechain types, these are the definitions of Chi1. Sidechains like arginine, lysine, glutamic acid, glutamine, all have a CG atom, a C gamma atom. Valine and isoleucine have a CG1 and a CG2. And so the definition of Chi1 is to the CG1 atom. There's a certain stereochemistry that tells you where CG2 is relative to CG1. Serine has an oxygen, hydroxyl, so it's OG, O gamma. Threonine also has hydroxyl, but it has a CG2, so because it has another heavy gamma atom. And so Chi1 is defined to OG1. And cysteine has a sulfur SG, so it's defined to the sulfur. For all of the 18 side chains, so alanine only has a CH3 group, and so it doesn't have a C gamma, it only has a C beta. So it doesn't have a Chi1 rotamer. And of course, glycine only has a hydrogen, and it does not have a Chi1 rotamer because it doesn't have C beta or C gamma. But all the other side chains do, and Chi1 is always rotameric. It is, with the exception of proline, it is always 3 different conformations at about plus 60, 180, and 300, or minus 60. Proline, because it has that tight ring, can only really kind of go like this or this, and so it's sort of plus 30 or minus 30, which is those two peaks here. You'll notice that for most sidechain types, the 300 one is the most common one, followed by the trans, followed by the plus 60. So the minus 60, G minus, trans, and G plus in that order. There are exceptions. Serine likes G plus because it can hydrogen bond back to the backbone when it's in that G plus conformation, because it's just short side chain with a hydroxyl. Threonine also likes that for the same reason in part. The side chains with 2 gamma heavy atoms, which are valine, isoleucine, and threonine, one of those rotamers basically always has a problem no matter what the backbone conformation is. And so you really see only 2 allowed conformations for threonine, and only 2 allowed conformations, one strongly allowed, one a little bit, and the other one not. And same thing for isoleucine. 1 allowed, 1 pretty low, and then almost nothing for the third one. And because of the definitions of where the gamma 1 atom is and where the gamma 2 atom is, these look a little different from each other. The definitions are kind of a little funky. Right. But when I say rotameric, I mean G plus trans, G minus. Some degrees of freedom in proteins are non-rotameric. They don't fall in those 3 peaks. They're much broader distributions, maybe only 1 peak, and really quite different than this G plus trans, G minus feature. So on the left-hand side here is arginine, where Chi1 is rotameric and Chi2 is rotameric. And so you see 9 different peaks or clusters here. 3 for Chi1, 3 for Chi2. First thing, and we can also note that populations are not equal. Some are really common and some are pretty rare. And I'm going to explain why they're rare in a little bit. But here's asparagine, where Chi1 is rotameric. You can see 3 groups of points, but Chi2 is not. You see, for instance, for the G plus rotamer, Chi2, G plus Chi1 rotamer, that Chi2 is this really broad distribution with a peak somewhere around here. Same thing for the trans rotamer. The G minus rotamer has 2 populations, very large one here and a smaller one here. But the distribution is like this. It's not 3 like that. This is stolen from Wikipedia. This is just pictures of all of the 20 amino acids. So as I'm referring to different amino acids, you may need to, if you want to look back at a couple of slides. So proline, for instance, which I've been gesturing to show you what proline looks like. I can find proline. Oh, there it is. This is this backbone N, C alpha, C beta, C gamma, C delta, back, or a ring back to the NH. The side chains with more than one gamma heavy atom are valine. So that's these two atoms here. Isoleucine, that atom and that atom, it has an extra one there. And threonine, which is in the polar right there, OH, and then CH3. The non-rotameric degrees of freedom occur because not all of the bonds in side chains are sp3 to sp3, tetrahedral carbon to tetrahedral carbon. Some of them are to sp2 carbons, which are flat, which have substituents that are 120 degrees apart, and the whole thing is in a plane. So let's look at an example of that. If we look at aspartic acid, for instance, this atom right here, this is the C gamma heavy atom. It's a carboxylate, and all of these atoms, this one, the C beta, the C gamma, the O delta 1, and the O delta 2, those are all in a plane. That's a flat group, and these are 120 degrees apart. This bond, and that bond. Same thing for glutamic acid, for glutamine, which is right here, and asparagine, they have an O and an NH2. And it's also true for aromatics. So if we look at tryptophan, this atom right here, that's the C gamma. That atom, that atom, that atom, that's 120 degrees apart. And the whole ring system is flat. Same thing true for tyrosine, phenylalanine, and histidine, which is hiding somewhere right up here at the top. So you can use this to kind of go back and look at particular, what are the atoms that make up. I have this stuff in my head because I've been looking at it for a long time, but I don't expect that all of you do. I like this diagram because it represents them as organic molecules with stereochemistry, and so that's what these, that's why this is sort of a wedge shape to show that it's coming up a little bit towards you. That's kind of the stereochemistry of this group. Okay. Taking a second because the next slide has a lot of dots on it. So here's what the distributions of some of the non-rotameric degrees of freedom look like. This is for asparagine, aspartic acid, glutamine, and glutamic acid. So the non-rotameric degrees of freedom are always the last one out. They're the furthest away from the backbone. You don't get rotameric, non-rotameric, rotameric again. They're all rotameric, rotameric, non-rotameric. So here's asparagine chi2, really broad distribution. It has some structure to it because of the different chi1 rotamers have different influences on this distribution. But basically, you wouldn't describe it as 3 peaks. And glutamine looks fairly similar. There's 2 populations there having to do with the chi1, chi2 rotamers. Aspartic acid and glutamic acid, because it's a carboxylate, the 2 oxygen atoms are the same. And so it doesn't matter whether you have this or this. And so we only plot chi2 from -90 to +90 because -90 and +90 are the same. Again, pretty broad distribution for both Asp chi2 and Glu chi3. And take a second for this slide to load. For the aromatics, phenylalanine and tyrosine are very similar to each other. Tyrosine just has an OH at the end of the 6-membered ring, benzene type ring. Almost all phenylalanines and tyrosines have a chi2 of about 90 degrees. They have a very small population at about 0. So, 90 degrees would be this, and 0 would be this. And that only occurs for the G- chi1 rotamer. The other 2 chi1 rotamers, trans and G+, do not have a population here. It's a very small kind of subpopulation of tyrosine and phenylalanine conformations. Histidine and tryptophan are a little bit more complicated. They do have different peaks. You would almost call them rotamers, but the distribution is still pretty broad. They can kind of, it's not as sharp as those ones that we were looking at before. There's Chi1 dependence to this, there's backbone dependence to this, and so it doesn't have 3 peaks in every situation. Like if you change the backbone conformation, you might see only one of these peaks and not the other 2. As I've been saying, even the rotameric, if we go backward, now we're going back to just the rotameric dihedral, the rotameric degrees of freedom. I already showed you that the Chi1s are not equal. The minus 60 is usually the highest, followed by the trans, followed by the G+. But if we take pairs of them, if we take, for this is arginine, Chi1 and Chi2 together, and I showed you a scatter plot of those 2 slides back, there are 9 possibilities. There were 9 regions. There were 9 places in that where there was a group of points together, clusters. They were not all equal. And here are the populations kind of laid out as a histogram. And it's clear that 1, 2, 3, 4 of them are really small populations. A couple of high ones and a couple of moderate ones. And so the question is, why are these populations not equal? And the reason is something that's called the Syn-pentane interaction. And if you take pentane, which is 5 carbons, CH3, CH2, 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 CH3, it has 2 degrees of freedom, 2 dihedral degrees of freedom. Atoms 1, 2, 3, 4, and then 2, 3, 4, 5. 2 sp3 type degrees of freedom that we've been looking at. And so they like G+ trans and G-, and they don't like to be in between those values. If we take all of the conformations that you could possibly have for pentane, the lowest one of all of those is trans-trans. It's when the heavy atoms, the carbons, are 180 degrees apart, and the hydrogens are plus 60, minus 60 degrees apart. And so trans-trans, we're going to call that energy 0. If I make one of the dihedrals either gauche plus or gauche minus, and I can make either the first dihedral or the second one, those molecules would all kind of look the same. The energy is about 0.9 kilocalories per mole higher. That's enough to make the populations like 70-30 or something between trans-trans and one gauche interaction, one G+ or G-. If I add 2 of them, if I have G+, G+ or G-, G-, where they're the same sign, then the whole thing kind of looks like this, and the first and last carbon are kind of far apart from each other. The energy is about 1.8, so roughly additive, roughly double what one of them is. And the reason is each one of those gauche plus interactions or gauche minus is worth about 0.9 kilocalories per mole, because the carbons are only 60 degrees apart. But the kicker is if the signs are opposite each other, it's G+, G- or G-, G+, it looks sort of like this, where the first and fifth atom are now really close together. They're so close together that, in fact, if you do try to make this molecule, or if the molecule sits in that conformation, the dihedrals are more like 80, plus or minus 80, rather than being 60. And if you take, if both angles were 0, you would start with a plane like that. And if you start to get to make one go in a plus direction and one in a minus, they move apart kind of really slowly, the first and fifth atom. And so this is a conformation that no sp3 type system wants to sit in. And so if we want to figure out, well, we have 9 conformations total, which ones are allowed and which ones are not allowed, what we want to make sure is that we don't have any G-, G+ or G+, G- combinations. And so Chi1 is N, there's a C alpha behind the screen here, C beta, and then C gamma. So this is going to be C gamma here, here, and here. And then the next atom out is C delta. And sort of trans is going to kind of, when it's trans, it's sort of coming towards you. G- is one direction and G+ is the other one. Kind of bending back into the screen, kind of towards where the nitrogen, the H alpha, or the carbonyl carbon would be. So we're just looking for the combinations where G plus G minus or G minus G plus occur. Those occur for when chi1 is G plus and chi2 is G minus. This delta atom is now too close to the nitrogen. It looks like that kind of situation. So that conformation should be forbidden. The same thing for G minus G plus, actual G minus G plus of chi1 and chi2. That's this one right here. But we also have to worry about those combinations of things for the carbonyl carbon. And so we have to do a little arithmetic that C, C alpha, C beta, C gamma is equal to chi1, which is N C alpha, C beta, C gamma, minus 120 degrees. It's just, around 120 degrees. And so if we do the math, it turns out that trans G minus would put a delta in a plus 60, minus 60 kind of situation. G plus would also do that here. And so those are the four combinations that are not allowed because the delta atom would just bump into the backbone nitrogen or the backbone carbonyl carbon. And it works. So if we look at the arginine, chi1, chi2 rotamer populations, calling them R1 and R2, rotamer 1, rotamer 2, the four that are not allowed, 1, 2, 3, 4, are the four that have almost no population. And so this Syn-pentane trick is a nice way to look at an organic molecule and try to work out, well, just in your head or on the back of an envelope, what conformations would be allowed and not allowed, at least locally, within a molecule. Leucine is a little bit more complicated, just to look at one other example, because it has two C delta carbons, C delta 1 and C delta 2. And so here's the chi1, chi2 plot. And a little bit hard to tell, but the populations of this one and this one are really high, and the rest of these are all pretty small. And the reason is, if you're going to do this kind of calculation, you got two C deltas to worry about in each of these situations, either one of which could have the G minus G plus problem. And so if you try to add up where you would have the G minus G plus problem, it would be 7 of the 9 conformations. Basically, all of the G plus chi1s are forbidden. You do see leucines that are fit incorrectly into electron density because they're sort of a little bit, they look symmetric. It's a problem. It's one of the side chains that's not always well determined in crystallography. One of the things I worked on when I was a grad student was trying to predict where all the side chains are in protein structures. Just give me the backbone coordinates, let's model all the side chains on them. And I was working on I was in Martin Karplus's group, and we were looking at the parameters for the CHAR molecular dynamics package, the parameters that are used as a force field to do simulations of proteins, molecular dynamics simulations of proteins. Looking at proline, the parameters that I was working on were proline. Proline is this ring and has sort of 2 conformations, kind of puckered up or puckered down. But those 2 rotamers are very strongly dependent on the backbone. If I move phi one way or the other, I'm either all up. If I move it the other way, they're all down. And I was working on sidechain prediction and thought, well, why don't I look at the phi psi dependence of the 3 rotamers, the chi1 rotamers, of all of the sidechain types, and see if there's any backbone dependence. Is something bumping into something? And I didn't expect it to have as strong an effect as proline does, but it has a strong enough effect to be really useful. So, for example, if I take the valine trans rotamer, and this is a phi psi plot, and the size of the box is whether all of the valines in that little 10 by 10 degree region, are they all trans? If it's a full-size box, that they're nearly 100% trans in that little phi psi region, 10 degrees by 10 degrees. Whereas the tiny little boxes means almost nobody is in a trans conformation. And you can see there's a pretty strong psi dependence this way, and that it's periodic, that you have a high population and a low population. There aren't many valines in here. Then a high population, 180 degrees up, and a low population. And it was pretty obvious that this pattern existed. For a while, I didn't know what it was until I was at a lecture by Stuart Schreiber, who was teaching drug design course and kind of mentioned this Syn-pentane trick. And we started to look at the backbone conformations relative to side chain conformations and whether that was going to help me figure out why does this pattern look the way it does. If we just look at the valine G minus and G plus, we see the same kind of thing. In this case, opposite trend, low population high, then low, and then high. And then the G plus is only really common in this upper left corner. So there's definitely, and there's a phi dependence on that one going this way. It's mostly psi dependence over here. So the question is why? And it's the same Syn-pentane sort of trick. There's a lot of arithmetic on here. I don't want to go through it in absolute excruciating detail. What happens is if you basically have to just connect 5 atoms. So if I start at the C gamma atom and I count 5 atoms, so C gamma is 1, C beta is 2, C alpha is 3, the nitrogen is 4, and the carbonyl carbon of the previous residue is 5. So I just have to check all of the possibilities of those 5 atoms having a G minus G plus or a G plus G minus type of combination. The same thing is true, and that's dependent on chi1, because that's where the gamma atom is going to sit. It's also dependent on phi, because phi kind of moves this carbonyl carbon around. So that's a rotation around these 2 atoms, and it would, sort of turn around like this. I can also count phi from the C gamma to the oxygen of the same amino acid. Gamma, C beta, C alpha, carbonyl carbon, oxygen, that's 5. And it's also 5 to the nitrogen of the next amino acid. And the position of those 2 amino acids is determined by psi. That's a rotation around this bond. So all of these equations up here are basically to figure out what combinations of phi and chi1 and psi and chi1 are bad. And those are listed here. So for instance, for the G plus rotamer, psi equals minus 60 is bad, and psi equals 120 is bad. This is the backbone nitrogen. This is the backbone oxygen. I may be the only person on the planet that could tell you that off the top of my head. Same thing is true up here. 0 and 180 are bad for the trans rotamer. And in fact, we saw that. Here's the trans rotamer. Here's 0 and 180. That's bad for the trans rotamer. And I won't go through all the possible combinations here. If we look at the population of the trans rotamer for valine, for instance, at 0, it goes way down like that. And again, at 180. For the G plus rotamer, it goes down at minus 60 and right around 120, plus or minus 30, 40 degrees. So it not only works conceptually to sort of look at these Syn-pentanes, but then it also works in the experimental data when you look at these kind of distributions. This is called the backbone dependent rotamer library, which is used in Rosetta. It's used in a lot of side chain prediction and protein design programs. And what it tells you is the frequency of all of the rotamers, not just the chi1, but chi1, 2, 3, 4, the lysine, as a function of phi and psi. So for every phi and psi on kind of a 10 degree by 10 degree grid, it gives the frequency of the valine trans G minus and G plus rotamers. And those numbers have to add up to 100%. And so here's just a plot of those. We redid it about 10 years ago now, specifically for Rosetta, because Rosetta takes the log of these and treats it like an energy function for the different rotamer conformations in protein design and protein structure prediction. And so we needed these surfaces to be smooth. And when I had done it 10 years before that, as a postdoc, I wasn't thinking about having a smooth distribution, and so I didn't worry about it. But we redid it for Rosetta so that these distributions are nice and smooth. So for instance, here's the trans rotamer again, with a high population that's actually at about plus 60, and this is at about plus 120, and it goes down to 0 at 180 and at 0 degrees. And remember that G plus was only really common in the upper left corner. It would be common in the lower left corner if there were many points down there, but there aren't. And then the valine G minus is sort of shifted from the trans by about 60 degrees. And so here are some of the publications, if you're kind of interested in this stuff. We also have on this second web page here, kind of all of this arithmetic to get the phi and psi and the chi1s to all line up, and the G plus G minus, if you're interested in that. Okay, the next thing I want to talk about are hydrogen bonds and protein folding. You might want to take a brief break now, be my guest, and pause the video and then restart, and this is kind of the next section. Proteins fold, and you've probably studied this in previous courses, because they're trying to take the hydrophobic side chains of the protein and bury those on the inside to get them away from water. That's why they're called hydrophobic. The reason that happens is water is very hydrogen bonded. You know, you've got H2O and two lone pairs on the oxygen, and so it forms this kind of lattice of every water molecule is interacting with four other water molecules nearby. If you've got a flat surface, or like a hydrophobic surface that can't hydrogen bond at all, can't make any kind of interaction at all, the waters all kind of want to interact with each other, and they don't want to interact with that surface, because if they point a hydrogen or lone pair at the hydrophobic surface, they're going to lose that hydrogen bond, which is a lot of energy. And there are only so many ways to do that, so there's sort of an entropic cost to solvating, putting water solvent on a hydrophobic surface. And so it's an entropic effect of why do hydrophobic residues want to get near each other. It's not that they're so attracted to each other, it's that the water molecules don't want to be pointed at the hydrophobic surface, they want to be pointed at each other. And so the water kind of escapes the surface, and two hydrophobic residues will get in contact, and there's a kind of a weak attraction, van der Waals attraction between, say, a phenylalanine and a isoleucine. Hydrogen bonds are a polar interaction, an electrostatic interaction between two polarized bonds. So a carbonyl, here is the carbonyl carbon and the carbonyl oxygen, is polarized. This is about plus 0.5 charge on the carbonyl carbon and minus 0.5 on the carbonyl oxygen. The NH of proteins, or OH and serine side chains, is also about plus 0.5, minus 0.5. And the way those want to orient themselves is plus, minus, in a row. They don't want to be minus, plus, minus, or plus, minus, plus. They don't want to be pointed at each other. There's a certain stability, electrostatic favorability to a minus, plus type of orientation. And that's a hydrogen bond. And so that's what we have here is plus, minus. The side chains can do it too. OHs, the carbonyl carbons, the NH2 carbonyl oxygens of like carboxylates, or the NH2s in asparagine. All of those groups can make hydrogen bonding interactions, either as acceptors or donors. Donor if you have the hydrogen, acceptor if you're the oxygen with extra electrons to spare. And they can do that with each other, side chain to side chain, or side chain to backbone. So proteins fold, they get their hydrophobic amino acids all in the center, but that's a lot of NHs and carbonyl oxygens would be buried in that process and taken away from water. Like if the protein's unfolded, all of those groups are interacting with the solvent, with water. To fold up the protein, you're taking those away from water and losing about 5 kilocalories per mole per hydrogen bond that you're losing with water, which is a bad thing. Because proteins have so many acceptors and donors, and proteins have evolved, the structures have evolved, so that if you're burying carbonyl oxygens or NH groups, they will almost always, like 95% of the time, have a partner, each acceptor, each carbonyl oxygen, for instance, will have an NH donor somewhere in its vicinity where you can make a nice hydrogen bond. And the place where that's most common is in alpha helices and beta sheets. Before I talk about that, I just want to talk one other point about this is the directionality of hydrogen bonds is really important and sometimes neglected in some force fields, molecular mechanics force fields. The carbonyl oxygen, for instance, has two lone pairs that are situated about 120 degrees away from each other and 120 degrees away from the carbonyl carbon. So all of this thing that I'm circling here is planar. That's a planar group of 1, 2, 3, 4, 5, 6 substituents. These are atoms, but these are just electrons that are kind of in a molecular orbital kind of pointing off in that direction. That means the negative charge is here, and the NH kind of wants to point at that. And so it sort of can't be in a straight line, NHCO. So it ends up being in this kind of bent configuration. And there's a 120-degree angle between those two places. And so if you're looking at a crystal structure of a protein, you can tell that the NH is typically, it doesn't exactly, this kind of angle, but they don't generally come in from this way. They generally come in from this angle. They can't come from on top either. If you see an NH pointing at a carbonyl group like this, it's not coming from above. It has to come in the plane. And I'll probably in the homework have you look at structures and say, is that a good looking hydrogen bond or is it not? Depending on which one you pick, you might get either answer. In crystal structures, you don't typically see the hydrogens or in cryo-EM. And so there's no hydrogen here, at least in the coordinates. What you see is the N and the O. And so that's the distance you can measure. And it should be about 3 angstroms plus or minus 0.2. A carbonyl carbon oxygen bond is about 1.2 angstroms. Carbon carbon bond is about 1.5. So it's about twice the distance of a carbon carbon bond. One thing to point out is that if this nitrogen was approaching this oxygen, let's say from above like this, it could not get as close as 3 angstroms. The van der Waals distance, how close the molecular orbitals want to get to each other, is more like 4.5 to 5, 4 to 4.5 angstroms. So for the O and the N to get as close as 3, there has to be this electrostatic interaction. There has to be something pulling them closer together than they would be if they approached from another direction than they would normally be without the electrostatics. Same thing is true of the side chains. So for instance, a hydroxyl is tetrahedral. It has a hydrogen, a carbon over here, and 2 lone pairs, and that's a tetrahedral arrangement. The NH has to approach along one of those tetrahedral positions. Proteins manage to fold, get their hydrophobics in the center because they can make all these hydrogen bonds. And the way to do that very efficiently, and these are favorable conformations anyway, but the hydrogen bonds kind of hold them together, is as an alpha helix or a beta sheet. These are the two main kinds of secondary structure of proteins. Primary structure is the sequence. The secondary structure is helices and beta sheet strands. Tertiary structure is the whole folded structure. Quaternary structure, which we'll look at later, is oligomers, several proteins in a large complex. The way alpha helices are formed, there is a hydrogen bond between carbonyl oxygen of residue I and the HN of residue I plus 4. And if you think about it, those are a little bit more than 3 residues apart because you've got carbonyl oxygen of residue I, NC alpha C of I plus 1, NC alpha C of I plus 2, NC alpha C of I plus 3 to the NH of I plus 4. So consequently, there are about 3.6 residues per turn in an alpha helix. So every time you go around 360 degrees, that's about 3.6 residues. The hydrogen bonds are kind of parallel with the direction of the helix, and the side chains kind of stick out along the sides. Beta sheets look like this, where you have a residue with its NH and its carbonyl oxygen pointed at the neighboring sheet strand with its carbonyl and its NH. The next residue is pointing the other way, these carbonyls and NHs. And so for this one, for instance, the first one is pointing upwards, but the second one is pointing downwards. And if there's a third strand, it's making the same kind of interaction with the third strand. And then the third residue along here, there it's pointed back at this top strand again. These are all anti-parallel strands. You can have both parallel and anti-parallel strands. And in large sheets, you can have combinations. You can have one this way, one backwards, one this way, one backwards. Almost any conformation is possible. And the side chains in beta sheets are one's pointed up, one's pointed down, one's pointed up, one's pointed down as you go along the strand. So these are things to keep in mind when you're looking at a structure. Kind of, if you look at a sheet, you know, half the side chains are pointing on this side of the sheet and the other half are pointing on that side of the sheet. So I'm going to show you this little movie. Oops. This is a simulation of, I've actually forgotten the name of the protein. I can add that link later. That I got from YouTube. It's TLR9, something like that. And what we're going to see is this is a molecular dynamics simulation of a protein, small protein folding. And you're going to see the hydrophobic groups in spheres. And they're going to try to be on the inside and in contact with each other generally. And you're going to see the secondary structure form. You'll see all of well, not all, but many of the side chains kind of wobbling around. And then, of course, there's music. Oh, NTL9, there we go. There's the name of the protein. So you'll hear them. So that helix is the first thing to form because it's local in the sequence. Here's the music. Hydrophobic collapse is when all the hydrophobic side chains get together on the inside of the protein, or most of them. So the secondary structure is mostly formed. Protein's not quite folded yet. There we go. So what it says is the hydrogen bonds have to be satisfied. Basically, when those Jane Richardson ribbons that describe beta sheets in the alpha helices, when those are present, then the hydrogen bonds are present, and vice versa. So we can stop that. Okay. The next thing I want to talk about are protein beta turns. So, between a helix and another helix, or a helix and a sheet strand, or one sheet strand and another sheet strand, are loops. They are irregular in the sense that they don't have repeating patterns of the same dihedral angles. In an alpha helix, you've got -60, -50 roughly for one amino acid, the next, each one in the alpha helix. For loops, they'll be in different regions of the Ramachandran map in what seems like a random sort of order. And long loops can be, a simple beta turn, a simple, beta sheet, turn, two residues that make up the loop, another beta sheet, or they can be longer. They can be 5, 10, 20, 50 amino acids that connect sometimes over very long distances, one piece of secondary structure and another piece of secondary structure, one helix with another helix. Protein loops do not look like this. They're not sort of nice, smooth objects like this. When you see them in PyMol or in the kind of ribbon diagrams, they look smoother than they actually are. What they really look like is they have a lot of little U-turns in them. So they look sort of like this. I call this my little Trump hand, little fingers. So the way to think of them is not like that, but really kind of, they have some internal structure to them that is dictated by the sequence, by certain interactions of some side chains with other side chains, or backbone and side chains with each other. So a major component, maybe I think it's about half of the residues in longer loops are in beta turns, which is why we study them kind of as a separate form of structure, not really secondary structure, but a kind of structural motif, is because they're so prevalent in long loops over 5 amino acids or 8 amino acids. The kind of classical definition of a beta turn is 4 residues that are not in an alpha helix, because they would look like, alpha helix looks like a succession of turns that are all the same. But if we look at things that are not helix, and the distance between alpha carbon of the first residue and of the fourth one has to be less than 7 angstroms, less than or equal to 7 angstroms. So the one that I'm showing here is 4.9 angstrom between this residue. So there's C alpha 1, 2, 3, and 4, and the distance between them is 4.9 angstroms. There are only certain ways to do that. There's only certain combinations of phi and psi and maybe omega of these middle 2 residues that would allow these two to get close together. You can imagine that a lot of conformations, the 1 and 4 would not be anywhere near each other. But there are certain places that are favorable conformations for residue type at 2, residue type at 3, such that you get this kind of U shape. And there's a classical nomenclature from this. Some of it also derived by Jane Richardson, and Janet Thornton is another person who's done a lot of the structural bioinformatics work in the 70s and 80s, long career in research, both Jane and Janet, who I look to as kind of mentors, and both of them still active, but people who've done things over many, many years. And we revisited some problems with much larger data sets and higher resolution data sets, but both of these women have done lots of work over decades that is, we're adding to the knowledge that they developed a long time ago. This sort of classical nomenclature is historical, and it was, Roman letters 1, 2, 3, 4, 5, 6, 7. Some of those turn types turned out not to be real. There were a couple of structures, maybe they weren't such good structures. So term type 7 no longer exists, it's not used. Term type 3 looks a lot like term type 1, and so it's not really used anymore. And so these are the ones that are still used. 1, 1 prime, 2, 2 prime, 6A1, 6A2, 6B, 8, and 4. So the type 1 is by far the most common. It's basically an alpha-alpha conformation, alpha region of the Ramachandran map. 1 prime is basically the mirror, it's not quite a mirror image, but sort of the opposite of that, where the dihedrals are opposite. Minus 60 plus 30 to 60 minus 30, minus 90 to plus 90. And so they're kind of variants of each other, in some ways look a lot like each other. 2 and 2 prime are the same way. This is a B conformation and an L, so BL. 6A1, 6A2, and 6B are all require a cis-proline at position 3. And so those are only relevant if this residue up here, this third residue, is a proline. Type 8 is an alpha to an A to a B region, AB type. And type 4 is everything else. So if you're within 30 degrees of all of these values, then you're one of these turn types. It's actually a 30 degree by 30 degree box, so it's sort of minus 15 actually in either direction. And some people use different. Like some people allow like 30 for the first residue and 40 for the second one, or vice versa. Type 4 is everything else. Any other kind of beta turn that doesn't fall into one of these bins, one of these little squares in the Ramachandran map, would be a type 4 turn, which is something like 30% of turns. And a couple of years ago, we started thinking about these and trying to update the structural bioinformatics of this, because we can do this on, excuse me, really high resolution structures, and kind of try to come up with a new nomenclature. So we did that by taking very high resolution structures at better than 1.2 angstroms resolution, better than or equal to. We used the dihedral angle metric, so we're directly comparing the dihedral angles of residues 2 and 3 and the omegas. And we use what's called the density-based clustering. We're looking for a lot of points near each other, separated by low density, and then another lot of points near each other. It's called the density-based clustering. It only puts points in clusters if they have a lot of other points around them. It's an algorithm called dbScan. And so this paper came out last year. If you really are interested, you can take a look at that paper. We wanted to come up with a nomenclature that was easier than Roman numerals with prime symbols. Most of my career, I could not have spouted this off to you. I guess now I can because we've been working on this, but most protein scientists could not tell you, what are the appropriate dihedrals for a type 8 beta term versus a type 2 or a type 1. So we wanted to name the turn after the conformation of residue 2 and residue 3. Those are the ones that vary. The omega of residue 2 places the C alpha of 1, and the omega of residue 3 places the C alpha of residue 4. So we don't need the phi and psi of residue 1 and 4. We just needed of 2 and 3 plus the omegas in between. And so we need a Ramachandran nomenclature for all the beta turns. We got 18 to preview it. We got 18 different turn types as opposed to the 7 or 8 in the previous page. So we gave names to all of the sub-regions of the Ramachandran map that have populations to them. The red dots are the average dihedral angles for one of the residues in our 18 turn types at either position 2 or 3. And so we needed different names for each of these regions. So there's a long history of looking at protein loops and dividing the Ramachandran map into smaller sections. So that big alpha helix region is, the alpha is at this end, that's where alpha helices are. The other end of it is called delta or D. B and P, we're just going to separate along this line, this sort of a lower density along that line. So the P's are here and the B's are here. I call this is polyproline 2 helix I mentioned earlier. This is that pre-proline conformation called a zeta in some papers. So we're going to call that a Z. These are gamma turns. I mentioned earlier the 3 residue turns. There's a few of them in that region, actually gamma prime. And what we did is we made the left-hand side all capital letters and the right-hand side all small letters so that it's easy to know which amino acids are on the left and which ones are on the right of the Ramachandran map. And they're the same letters if you go through the origin. So capital A to small a, capital D to small d, capital P at this far end to small letter p down here. So just go straight through the origin in a straight line. And generally something on this side has a counterpart on this side. There's no counterpart of Z's. There's almost nothing in the lowercase g. We had to come up with one more letter for this region. Jane Richardson was our reviewer, and she was a great reviewer. She had really useful comments. One of them was, well, we have this thing here. We needed to give it a different name. And I don't know why she wanted to call it N, but she wanted to call it N. So we called it N. So when we do this kind of clustering on, I think it's about 13,000 beta turns in very high resolution structures, this is what we get. We get a lot of the same turn types back, we would expect to. So we get type 1 back. And we're going to name that capital A, capital D, because this is the first residue is in an A conformation, or residue 2 out of 4 is in an A conformation, residue 3 out of 4 is in a D conformation. So it's a little bit northwest of the A conformation. So that's capital A, capital D. Type 1 prime, remember the prime ones are all across the map through the origin, is going to be lowercase a, lowercase d. And you can see this, and this is the sort of sequence logo, the frequency of each of the 20 amino acids at each position. And this one, type 1s pretty much require a glycine at position 3. They like glycine at position 2, but they also like asparagine and aspartic acid and some serine. Here's type 2 and type 2 prime. Type 2 we decided to divide into 2 groups because you actually plot them together, they're kind of there would be 2 peaks in here. So we separated them into this one and this one. And they have different frequencies in the 3rd residue position. This one really has to have glycine. This one, glycine's okay, but it really prefers asparagine, aspartic acid. Remember, those have more left conformations when I was showing the Ramachandran plots way earlier in the talk. This is type 2 prime, which has to have something down here, which really can only be glycine, very small percentage of other amino acid types. We got some new types that hadn't really been named before. So small d, capital D, so basically a left-handed helix region to right-handed helix region. Capital D, small d, left or right-handed helix region to left-handed helix region. And so we got, I think, 6 or 7 different new types. Just to give you a summary of it, here's a table with the percentage. So type 1 AD turns, previous name is type 1. That's what we hope will become the previous name, is almost 50% of all beta turns. And here are the average dihedral angles. So some of these are, subdivisions of previous turn types. Some of them are new. Some of them require cis proline either at the at the third position of 4, and a couple at the first at the second position of 4. You can read through this, and so this is all in this paper. If you ever want to look at the beta turns in a structure, there's code to Python script to assign them. Okay. Now's a good moment to pause. I'm going to take a couple-minute break, and if you pause your video, when you come back, I will continue, and we're going to talk for maybe 15, 20 minutes about biological assemblies. So I'm just going to pause the recording and hope I don't mess it up the way I did earlier today. So come back in a few minutes. Okay, now we're going to talk about biological assemblies. We've been talking about atomic interactions, side chain conformations, dihedrals, very local pieces of structure, beta turns. Now we want to talk about quaternary structure, which is interaction of proteins either with other proteins or other molecules like ligands and nucleic acids. Most proteins and most structures in the PDB are of oligomers. Some proteins act as monomers, say enzymes, and they act on small molecules. But a lot of enzymes work as homodimers or tetramers or octamers, multiple copies of the same protein type, same sequence, usually in a symmetric kind of complex. So in the middle here, we have guanine monophosphate reductase, which is an octamer. On the left-hand side, we have a complex of 2 different proteins, heterodimer, peroxidase and cytochrome C. And on the right-hand side, we have a nucleosome, which is 2 rounds of the DNA wrapped around a set of 8 proteins of 4 different types, which is the histone octamer. We can see if you do cryo-EM structure, you kind of get, or an NMR structure, you get the biological assembly, which is kind of the most biologically relevant object complex observed in the experimental data. You get that kind of for free because you're not making a crystal out of it, and so you're not getting crystal contacts that are not biological that help hold the crystal together. You're getting just the interactions of the complex or whatever it is that you purified and frozen in cryo-EM. Crystals, on the other hand, if a protein is a homodimer in solution or in cells, but in solution when you purify it, make it, it'll enter the crystal as that dimer, but then the dimers will make lots of contacts with other dimers that are not biological. They're only there because you made a crystal out of it. And so it's an interesting and important problem of, if you look at a crystal, what's the right dimer or what's the right octamer, or is it a dimer or is it an octamer? So if we take an example, this is human estrogen sulfotransferase. It's known that it's a dimeric protein, but here's the crystal, which dimer is it? And that's the problem of finding the biological assembly within protein crystals. To understand this, we need to think about how crystals are constructed and how they're thought about by crystallographers. When a crystallographer is trying to model the data, model the experimental data, those spots in the diffraction pattern, the way they do it is to think about, to figure out what is the smallest object that I can use and build coordinates for, and then copy it many times and use rotation and translation to build up a model of the entire crystal. And so that smallest unit is called an asymmetric unit. It might be just one monomer of the protein. It might be two monomers of the protein. It might be a heterodimer if it's two different proteins. There can be different ways to model the same crystal. But typically, it's sort of whatever amount of symmetry exists in the crystal, the asymmetric unit is kind of the smallest bit that would successfully model the experimental data or would model it the best way. And so at the top, we can think of the asymmetric unit as just this little arrow. That's the smallest unit we might use for this crystal. And then we can build up what's called the unit cell, and that's by symmetry, basically a set of rules that says if you have one of them, you can build another one by just rotating it around an axis, and now you have another one. So this unit cell is two monomers. Typically, there are 4 or 8 or 12 or 16 operators that help you build up the unit cell. And the unit cell is either a cube or an elongated cube where the lengths are not all the same, or even a parallelepiped where some of the angles are 120 degrees instead of 90 degrees. But it's a kind of object with a cube-like shape. I'm just watching all the Star Trek Picard, so you like a Borg cube. It's a really good series. The way you take asymmetric unit gives you the unit cell. Once I have a unit cell, I can then make copies of the unit cell and build them up in 3-dimensional lattice. So along the x-axis, y-axis, and z-axis, and build up a model of the entire crystal, which ought to explain the experimental data. Now, in this case, if I know my protein is a homodimer, I have to look at this and think, well, which homodimer? Is it the one inside each unit cell? Or is it the one between unit cells? You have to pretend that these little arrows are not symmetric, and that this dimer, this purple-green dimer, would be different than this green-purple dimer. Or there are molecules like actin that are actually polymers. We get many copies kind of attached to each other. So maybe all the purple arrows in a stripe could be a biological structure. That's not common in PDB structures, but there are structures like actin where that does exist. So, just as an example, if we look at hemoglobin, hemoglobin is a tetramer of 2 different chains. So we call it an A2B2 tetramer. That's the stoichiometry. So 2 of alpha hemoglobin chain and beta hemoglobin chain, which are homologous to each other, but not the same sequence. They're different genes. So the first one on the left is the asymmetric unit of this PDB entry, 2HHB. PDB entries always start with a number from 1 to 9, and then followed by 3 letters or 3 letters and numbers. So this asymmetric unit is hemoglobin, is exactly what hemoglobin is. It's got 4 proteins, 2 copies of each. That's the biological assembly. So in this case, the biological assembly is the same as the asymmetric unit. That's true for about 40% of crystals in the PDB. The middle one, the asymmetric unit, is this heterodimer. It's only half of the biological assembly. So in order to build up the biological assembly, we have to duplicate the coordinates of this and rotate them around with a symmetry operator and build something that looks like this. So in this case, the biological assembly is bigger than the asymmetric unit. If you looked at the crystal, like this crystal, and you built it up, you would actually see that it is made of objects that look like this. It's just that when the crystallographers decided to solve the structure, they found that they could model it with just one of these. And so it's like, how much experimental data do you have to how many parameters that you need to fit? So the parameters you need to fit are the coordinates of all of the atoms. And so if I can do that with only 2 proteins instead of 4, I get a kind of higher signal to noise ratio, or higher experimental data information to parameter that I need to fit. So that's why the goal is to use the smallest asymmetric unit that is needed. That's about 30% of crystals, where the biological assembly is actually bigger than the asymmetric unit, and you need to build it from more than one copy of the asymmetric unit. And finally, the biological assembly can actually be smaller than the asymmetric unit. So 1HV4 actually has 2 copies of hemoglobin in the asymmetric unit. There must have been a little bit of asymmetry between these, or something to do with the lattice symmetry of the crystal, that the only way to model it was really to model it as 2 copies of hemoglobin. There's no reason to think that 2 molecules of hemoglobin belong together and that that's a biological interaction. It's just that was the way to model the crystal. So the biological assembly is actually smaller than the asymmetric unit, and that happens for about 30% of crystals, crystal structures in the PDB. We can think of biological assemblies as stoichiometry, which is how many of each type of protein, interfaces, how those proteins all interact, and symmetry, these 3 different aspects of a biological assembly. For the stoichiometry, you can represent those with a very simple algebra, which has each different type of protein sequence is a Roman letter, A, B, C, D. And then we just put down how many copies of each of those there are. So a monomer is just represented as A. It could be A1, but it's usually just written as A. A homodimer is A2, a homotrimer is A3. A heterodimer has 2 letters in it, so it might be AB. It would be AB. A trimer would be ABC, heterotrimer, 3 different substituents. You could have a heterotrimer that has 2 copies of A and 1 copy of B. We call this an uneven stoichiometry because there's not the same number of subunits of each type. That doesn't happen that often, but it does happen. You can have at least 2 different kinds of heterotetramers. One would be ABCD, where they're all 4 different sequences, 4 different proteins. But then you can have, like hemoglobin, just 2 copies each of 2 different proteins, and we call that an A2B2 heterotetramer. If you're looking at a PDB structure on the PDB's webpage for that structure, it'll tell you the stoichiometry of the biological assembly that the authors have deposited. It will also tell you the stoichiometry of the asymmetric unit, and it will do it in this format, sort of A, A2, A3, etc. Now we need to think about, we thought about the stoichiometry, we need to think about the interfaces. The interfaces can be either, if they're the same protein, 2 copies of the same protein, they can be either isologous or heterologous. Isologous means they're symmetric. So take one protein, rotate it about an axis, and get another copy of the same protein. And all the contacts of one protein looking at the other copy look the same as if you look at the second protein looking towards the first one. So in this crystal, there are 2 different isologous interfaces. One looks like this on the left-hand side, and one looks like this. This turns out this protein is a biological dimer. I think it's a kinase. Where this is the biological, actually I know it's a kinase, it's a BRAF kinase. This isologous interface is the homodimer that is biological. This isologous interface in this crystal is not. You can tell it's kind of loopy. There's not as much surface area involved in the one on the right as there is on the left. This is just a crystal contact, but it is symmetric. These other 2 interfaces are not symmetric. They're not, the residues on this side of the interface are not the same part of the protein that's on this one. Same thing over here. And it's important when we look at a crystal to try to figure out what's the biological assembly, of knowing what all the unique interfaces are and then knowing what their symmetry is. Are they symmetric or not symmetric, isologous or heterologous? So for vast majority of structures, if it's an oligomer, if it's a homooligomer in particular, the structures are symmetric in some way. So, protein could be just a monomer, but if it's a homodimer, it is very typically a symmetric homodimer, isologous interface. And we're going to name all of the symmetries of biological assemblies, beginning with the letter C or the letter D. I'm going to talk about the D dihedral symmetry in a minute. But if we're looking at the C's, this is called a C2 homodimer because it's cyclic. There's a rotation around an axis going through between the 2 proteins going into my screen, the red dot. And it's C2 because it's 180 degree, 2-fold rotation. If I just rotate this 180 degrees around, I get basically an object that looks the same. If I look at a homotrimer, on the other hand, each of these is rotated 120. If I just rotate this whole object around 120 degrees, I would get the same object again. The interface between monomer 1 and monomer 2 is heterologous. It's asymmetric. And it's the same interface between 1 and 2, and 2 and 3, and 3 and 1. In all three cases, it's the same interface, but they're heterologous interfaces. Because each one is sort of rotated like this, and so the contacts are different. If they were the same, you couldn't build up a C3 homotrimer with isologous interfaces, because where would you put the third one if you're already taking up the interfaces in the homodimer? So these have to be heterologous. Same thing in this homotetramer. Just rotate each one around, rotate this around 90 degrees, and you would get the same object rotating around that dot. And the same interface, 1, 2, 3, 4. The interfaces across don't matter too much. They're smaller, in the ones around the circle that connect are enough to define this, this assembly. The other kind of symmetry is called dihedral symmetry. And as an example of dihedral symmetry, this is going to be a D2 tetramer. And the way you build up a D2 tetramer is you take a C2 homodimer and another C2 homodimer, and you put them face to face, not back to front like this, because that would be actin, where you would have one, two, a filament. This is going to be face to face, and that's a closed assembly. You can't add anything to it that would be the same. So here's one C2 homodimer, here's another C2 homodimer. If you put those together, and it sort of doesn't matter for this particular complex, it's got a particular orientation of those two, but for other proteins, it could be this way, it could be this way, just any combination depending on what the proteins are. And we call that a D2 symmetry structure. And it's a homotetramer in this case. We can put together any pair of identical cyclic oligomers face to face and make them into a dihedral symmetry that has twice as many subunits. So for instance, this is the other two, this is kind of breaking up this cluster into the yellow and orange monomers, those two and those two. It's just a different way of breaking these two up. Remember, these are isologous interfaces because they're homodimers. What ends up happening is you end up having isologous interfaces between any protein in this one and any protein in this one. So the blue to magenta looks like this. And you can tell that's symmetric and isologous. So if you put the two cycles together, in dimers, each of the cycles is the C2s are isologous, and the interfaces between the two C2s are isologous. But we can build up larger ones. This is a C4 tetramer, so cycle, you can see 4 proteins here in a cycle. The same C4 tetramer, now we're going to put them face to face, and we get a D4 octamer. So the interfaces in these are heterologous because they're 1, 2, 3, 4 run in a circle. But the interfaces between any protein in the top and any protein in the bottom, those interfaces are all isologous. They have to be symmetric. You can't do it in some weird angle or something. At least that's not what typically happens for the vast majority of proteins. All of the interactions, each protein in the top is making an interaction to one protein in the bottom in a similar way. So just as an example, if I take the blue protein in this one and I look at its interaction with each of the 4 proteins in the top cycle, they look like this, 1, 2, 3, 4. These two are not really in contact, but just for the orientation with respect to each other is isologous or symmetric. And so that's true of every protein. Every protein has one of these and one of these and 2 of those in this particular assembly. We can advance this to heterooligomers. If we take 3 heterodimers and put them in a cycle 120 degrees around, we get a C3 trimer that has stoichiometry A3B3. So it has, if you consider each of these a single object, like just make it's two different proteins, but we just consider it the two proteins as one big protein, even though they're not covalently attached, and make that the object. So it still has a C3 symmetry. It has heterologous interfaces between each of those heterodimers. I mentioned that some crystals have filamenta and some proteins have filamentous type organization. So actin is a series of monomers with a couple of different types of interfaces. And you see it, here's a section of 8 copies of actin. The one at the top is interesting because it's inosine monophosphate dehydrogenase, which is a D4 octamer, so nice close like that. But it turns out, if you look in micrographs, you can see IMPDH in cells making this kind of filament structure. And in some of the crystals of IMPDH, you can see it. It's basically back-to-back copies of this D4 octamer, and it makes kind of a filamentous type of structure. You can have, I mentioned earlier, you can have an A to B type stoichiometry for a trimer, kind of weird stoichiometry, uneven. So 2 copies of protein A and there's 1 copy of protein B is attached. So here's just an example of that. One way to look, the question is, if we look at a crystal, how do we know what's the right biological assembly? And there are several different ways of doing that. One way to think about it is, if there are many structures of the same protein, let's say the protein's a dimer, and there's this one interface that is in every structure of that protein, and maybe other protein family members that are not too distantly related, and they all have the same dimer in every single crystal, but all the crystal contacts are different if the crystal symmetry is different. So you begin to think, well, that dimer is probably the biological one because we've seen it over and over again. It's like anything in evolution. If you see it over and over again in many different branches of life or something, it maybe ancient, goes back to much earlier organisms that then branched into multiple different organisms. That's a really bad description of evolution, but there we go. So for instance, if we look at this one of C up here in the upper right, that's this crystal structure 1B8X is one of these, but we see the same dimer interface in many different crystals of this protein and related proteins. You can see it also for heterodimers, where this is an RGS, the regulatory protein for G-coupled protein receptors, GPCRs. The G-alpha protein binds to RGS proteins that regulate it. And this heterodimer is seen in many different crystal structures of when these two proteins are present. And so we use that. I'm going to show you in a bit our website called PROTCID, Protein Common Interface Database, but we use this to try to determine, guess, correct the annotations in the PDB. In some cases, it's a hypothesis. It may be not well known or well studied. In other cases, it is well studied, but some of the entries are annotated incorrectly, so we can use PROTCID to looking for these common interfaces to clean up bad annotations. About 20% of the entries in the PDB have the wrong biological assembly annotated, maybe between 10 and 20% of crystal structures. And that's because the authors didn't see it, didn't deposit it correctly. They knew what it was. They wrote about a trimer, but they deposited a monomer as the biological assembly. So sometimes it's just a communication error. In other cases, they just didn't see it and didn't pay attention to it. We can use PROTCID to try to help us get the right answer. So this is a case of EGFR, which has a very unusual asymmetric heterologous homodimer that's part of the regulation of EGFR. EGFR has extracellular domains are the receptor, transmembrane helices, and then an intracellular kinase domain. And to activate the kinase, one of those monomers interacts with the other one, C-terminal domain, where my palm is, to N-terminal domain, where my fingers are, in a kind of asymmetric fashion. We see that in a lot of different crystal forms and a lot of different PDB entries of EGFR. and then also of its fairly close homologs of ERB2, ERB4, and ERB3. ERB2 is the HER2, same protein is called HER2, which is overexpressed in some breast cancers, and trastuzumab or Herceptin is an antibody to the extracellular domains of this protein. So we see this in a lot of different crystal forms, and John Currian, a long time, 15 years ago, identified this asymmetric homodimer as part of the activation of EGFR kinase domain. If he didn't do that, we would look at it today and say, oh, look, this homodimer is in many, many different crystal forms of some related proteins. Maybe there's something biologically interesting about it. Another way to use that is there are some homodimers or larger oligomers that are in hundreds of different PDB entries and dozens and dozens of different crystal forms, like these PALP ones. It's a very stable homodimer. It's very common. It's very well known. 10% of the entries might be incorrectly annotated, but we can see the same dimer in those entries as in the other 90%. And we can fix those annotations by this method of looking for the same interface in many different crystal forms. Here's another case of a kinase and cyclin. This is a heterodimer interaction. You see it in many, many different structures. But if the assemblies are kind of weak or transient, people might look at the crystal and go, I don't know if that's a dimer, or I don't know which dimer it is. Maybe they'll annotate the wrong one. We got in this business of looking at these same interfaces by these sulfotransferases. And there was a paper about 20 years ago that did cross-linking of estrogen sulfotransferase that I showed you the crystal earlier. And it was a very small interface that was cross-linked, and then they did protease digestion and mass spec. And at the time, a colleague was interested in that protein or a homolog of it. And we looked at all the different crystal forms of proteins in this family, and they all had this same small dimer interface. And none of the programs out there that do this kind of annotation would make that the biological dimer because the interface was too small. But there's really good experimental evidence for it, and it's in all of the crystal forms of these related proteins. And so we took that as a suggestion to go do it for the entire PDB for every protein family. And so, this way of looking, this is how you would generate a hypothesis. If you didn't know, you didn't have the cross-linking data, and you observe this dimer in many different crystal forms of related proteins, you might begin to hypothesize that that's the experimental dimer, and you could then decide to do experiments where, for instance, you mutated residues in that interface and experimentally determine that that protein is no longer a dimer. Another example of that is RAS. HRAS, KRAS, and NRAS are thought to behave as dimers or perhaps even larger oligomers. They bind to dimeric proteins like RAF kinase. But what the correct dimer has been controversial. And we have in PROTCID, in our common interface database, a dimer that has the alpha-4 and alpha-5 helices, the two C-terminal helices, in a nice symmetric isologous homodimer that's in many crystal forms of KRAS, 3 of HRAS and 1 of NRAS, and the dimer is basically the same in all of these cases. Since we first observed that, and we were adding it to a paper on this thing, there's experimental data actually for all 3 proteins that is a biologically relevant homodimer of RAS, which has a lot of implications for how RAS acts in cancer. It's very commonly mutant. It's like 90-something% of pancreatic tumors have a mutant KRAS, I think, or HRAS, KRAS. And so knowing how it dimerizes is knowing how it functions, and may help us think about drugs or certainly mechanism of how RAS is involved in cancer. That dimer is consistent with some of the RAS effectors, other proteins that bind to active RAS, GTP-bound RAS. They can bind at the same time as the dimer forms, and in fact they do in some crystals where you've got the RAS effector domains. Say RAF1 kinase has called the RAS effector domain that is this magenta protein and makes a heterotetramer, A2 heterotetramer in the crystal of RAF1 kinase effector domain with HRAS. And finally, well, almost finally, we wrote up a paper on this. I need to add the link. I will do that. In current opinions in structural biology, where a lot of these images came from, this is just some statistics about the different kinds of symmetry that are observed in crystal structures. So diffraction means any kind of X-ray crystallography or neutron diffraction on crystals. Most entries are C1 symmetry. That includes monomers, but any heterocomplex of different proteins, A, B, C, D, large assemblies would be C1. So it's sort of not it's called a cyclic symmetry, but it's basically not a cycle because there's only one object. C2 is 27%, and then everything else is pretty small. There are a lot of D2s, well, 5% D2s, and some other kinds of symmetry down here. NMR are typically monomeric proteins. There aren't very many dimers or oligomers. Electron microscopy, the idea really is to do larger assemblies. It's better at larger assemblies. And a lot of those are C1 because there are many different proteins together in assembly. But there are some viruses that are icosahedral. The SARS-CoV coronavirus 2 there are cryo-EM structures of that spike protein now. Or helical, which basically helical ones are these filamentous type things. So there's just some data on what's in the PDB. The last thing I want to do is there are a couple of different websites that try to look at a crystal and tell you or do an assessment of what it thinks are the correct biological assemblies. One of those is PISA, which goes back, I think about 20 years, which looks at biophysical features. It looks at the surface areas, hydrophobic interactions, hydrogen bonds, entropy of the assembly. It's better to have separate proteins, but it tries to figure out how much entropy would you lose in making a larger oligomer, and symmetry. It's looking for symmetric, cyclic or dihedral symmetry of the assemblies. It won't give you an asymmetric assembly typically, largely because those are not common, and many of them would lead to these kind of filaments. Again, not common. If you know your protein's a filament, you can look at your crystal that way. But if you're looking at some enzyme structure, it's not likely to be that kind of a filament. EPIC is kind of similar as a newer program that uses sequence conservation between the interfaces to figure out, well, which interfaces are biological, and then use the symmetry of the assemblies that you can build, again, cyclic or dihedral, and then kind of evaluates all of the different interfaces in those assemblies to figure out, is it a dimer or a tetramer or something larger. And then there's PROTSID, which we really only do at the interface level. We're looking at it now at the assembly level, but right now it's at the interface level. To just see if the same interface is present in many different crystal forms. We cluster all of the interfaces in all of the crystals of each protein family. So I just want to very briefly look at a couple of those web pages. If I look at EPIC, and I'm going to put in this protein 5HNR, that's the PDB entry. This is an octameric protein. The asymmetric unit is a monomer, but if you use symmetry operators, you can build up an octamer. So if I put in 5HNR and I just hit the submit button, the first assembly I listed up here has 8 copies of the protein. It says 8 right there. Get a little bigger. This is kind of all of the interactions as done as a graph. It says over here that there are 4 different interfaces. They're predicted to be biological, and that this assembly that it built up is what the PDB thinks is the biological assembly. So it gets a little gold star. Actually, everybody gets a gold star. That's not fair. It gets the logo PDB. Whereas these dimers may be part of the larger assembly, or they're just observed in the crystal, but they're not what the PDB thinks is biological. And you can click on the 3D view, and you'll get the protein to rotate around. The other one is PISA. This is the biophysical one. And if I hit the button that says analyze, it will say most probable assembly is octamer, 8mer. And if I just hit the assemblies button, it'll load in a second, I will see whether it what the assembly is listed. And the first assembly is A8, meaning that's the stoichiometry. The small letters are the ligands, the zinc sulfate. So A8 is an octamer. If I hit the download button with that checked, I would get the coordinates for the octamer and I could look at it with PyMol. It has a delta G that's -500, which is a pretty large value for the octamer, whereas this dimer, whichever dimer that is, maybe a crystal dimer, is only -100. So that by far is this much stronger prediction for that one, for the octamer, than it is for the dimer. And then finally, we can look at ProtSid. Reload this. Come on. And you just put the PDB entry in there and hit the submit button. These things all kind of work the same. And it's going to show us some information about that particular PDB entry. Here's the sequence. The PFAM is the family. What protein family is this protein in? It's called ALAD is the name of that domain. And we're going to look at the interactions between full protein chains. This is chain based. We can also, if this was a multi-domain protein with like a kinase domain and SH2, I could look at kinase SH2, or SH2. But this protein only has one domain, so I might as well look at the whole chain. And I'm going to hit this button that says retrieve common interface clusters. And what the page tells me is for ALAD, this family of proteins, this PFAM, there are 9 crystal forms total. That's what's available inside the protein data bank. It's 46 PDB entries. And then we've clustered all the interfaces in all of those crystals. And the clusters are numbered 1, 2, 3, 4, 5 by the number of crystal forms, the most to the least. So the first couple of interfaces are in all 9 crystal forms. That's 100%, 1.0 of all 9 of them. And the biological assemblies, the interface is kind of, there's a picture of it over there. In 98%, 42 out of 43 of those entries, this dimer is part of the assembly, is part of the biological assembly. So I can click this plus sign, and it would open up a table about all of the PDB entries that are in this cluster. And you can see the space group. This is the crystal form. We just give them a number, 1, 2, 3, 4, 5, 6, 7, 8, 9. We group all the ones that are the same crystal form. These basically, all these PDB entries have the same crystal. And is this interface in it's in PDBBA? I mean, basically, is this interface in this cluster in the biological assembly? And 1 means yes, and 0 means no. So this PDB entry 2Z0I, the biological assembly, the stoichiometry is A2. They didn't build an octamer, even though that octamer is probably present in the crystal. So that's probably a mistake on their part. And there's other information about what protein it is, the species, all of that. You can see there's actually several different interfaces that are in most of the crystal forms. And that's because this is, hold on a second. There we go. This is an octamer, so it has a number of different interfaces. And each of those is present in all of these crystals of all of these proteins. If I hit this button here is a link, this number 1, and it's going to download a file. Targ is a kind of an archive. If you just double click it, you will get a folder that has the coordinates of all of these interfaces, of all of these homodimers. And if I just hit one of these PyMol scripts, it's going to run PyMol and open up all of those structures. And color them from blue to red, from N to C terminus. And you can see this is a dimer that is present in all of these crystal structures. And you can click them on and off. I'm gonna, in a video later today or tonight, I'll show you how to use PyMol and how to manipulate it. But basically, this dimer is in all of those crystals. If we did number 2, we would find exactly the same thing. We would find a different dimer that is in all of the crystals. And if I just very quickly do that. When I was showing the D4 octamer a few slides ago, that's this protein. That's a PDB entry like this. So here's one of the dimers. That was, this one's isologous, so it's a dimer between one of the cycles and another one of the cycles. Go back to the PowerPoint. That's all I want to tell you about. The other part of this is going to be just how to navigate the PDB's website. Maybe you've done that, and to show you how to use PyMol. There are a lot of tutorials of PyMol on YouTube, so I'm going to try to do a kind of short presentation, and maybe I'll give you some links to some longer ones. So this went on a bit longer than I thought, but because I don't have a captive audience in a classroom, you can just keep talking. So feel free to email me if you have any questions. I'm happy to also do a video chat or a phone call or something if I can answer any questions. And that's it. We will send you the link for this video and for the next video. Take care and stay safe.